use the microphone. So welcome, I'm Dr. Villegas. This is the first class of 395 that we're offering biochemistry for the life sciences. So, what are we doing in this class, right? So, we used to teach a different class. There's a normal lecture class. And what we found was, as we expected, nobody wants to be there. Nobody wants to be here either, I'm sure, right? Why are you taking this class? Right, somebody. You. Why are you taking this class? Yeah.
As a requirement for med school.
Why are you taking this class?
Me.
Yeah. Either one.
Requirement for grad school.
Because it's a requirement, right? It's a requirement for med school, it's a requirement for grad school. Why is it a requirement? Right? Why do you have to take this godforsaken class? No one knows, right? So that's what we revamped the course to answer, right? Why are you here? Why does this matter? Right? Very few of you are going to go on to be biochemists, if any, right? Most of you are going to medical school, some of you are going to graduate school, some to nursing school, what have you. So the point of this class is going to be to revamp the way that we teach this class, right? What we found was us talking at you for 80 minutes just drives you guys crazy, drives us crazy because it gets our throat sore, and you guys don't want to be here. By the third week, we're having about 20% attendance. Frankly, it was not great, right? So why is it that you're taking this class? Why do you care? So that's what we're hoping to accomplish here, right? So what I want to run this class as is more kind of like as an office hours, right? Why do we want to run it that way? What we found is that students that come to office hours, right, they come with questions, you go over homework problems. I don't care if we go over all of the homework problems and get all of the answers here, right? What matters is that you take the topics that you learn about in class and in the book, and you learn why they're important, how to apply them, and why they're going to matter to you as you go on to medical school or graduate school or nursing school or whatever, right? I don't know how messy it's going to get, so you guys get to be my guinea pigs this semester, right? Hopefully it'll be fun. Let's go over kind of the structure of the classes and the structure of the course, okay? Your grades are going to consist of 50% exams. There are 4 exams, meaning each exam is worth 12.5% of your total grade. These exams are going to be given during our normal class times with the exception of the last exam, exam 4, which is going to be given during our final exam period. It's not going to be a cumulative exam. It's going to be just the 4th exam covering whatever topics the 4th exam would have covered, right? So 4 exams. 25% of your grade is going to be your homework assignments. And the homework assignments are going to be due the day before the exam that covers that chapter. So say chapter 11 is covered in exam 2. Chapter 11 of homework is due the day before exam number 2. Okay, so you don't have to worry about getting the all done on Sunday. My recommendation, what I strongly urge you to do, is to not wait until the day before the exam, because what's going to happen, you're going to have no idea what you're doing, right? You're going to lose time that you could be otherwise spent studying, right? So my advice would be to do the homework as we go, right? Of course, that's going to come down to the class structures as well, as we'll go over in a minute. 15% of your grade is the quizzes. The quizzes will be due before class. So I've set them all up to be due at 2 p.m. whatever day we're covering that chapter. Except for today, obviously, there is no chapter 1 quiz. It's going to start with chapter 2, right? So next Friday, you're going to have your chapter 2 quiz due before class. What this means is that I expect you to read the chapter before class, right? And why is this? Because again, what we're doing here in class, the class structure is going to be about half an hour of questions from you guys and lecture. Again, except for today. Today we're just going to go over some basic concepts, and then we'll start this off on Friday. But the first 20 to 30 minutes of the class is going to be questions from you and a short recap of topics that I've found in the past students have the most problem with, right? So I'm not going to be doing a full recap of the entire chapter and talking to you for 80 minutes. The first part of class is going to be for you to ask questions about material that you've read already that you're not sure about. Anything that I know a lot of you are going to have trouble with, we'll go over during lecture, right? During that first half hour. After that, I want to open the floor, A, to questions that you have about the homework, and also practice problems that I might come up with, right? Things that will illustrate the topics that we talk about and that we learn about in lecture, right? So today we're going to go over some practice problems that I've come up with. You guys haven't taken a look at the homework yet, obviously. Going forward, take a look at the homework first, right? Why do you want to look at the homework first before you come to class? Yes, the quizzes are going to make sure that you read the chapter, but why do you want to look at the homework before you come to class?
Do you have questions you might have?
You know what questions you might have. What happens if you have questions during class on the homework? Ask me, you might get the answer. I don't care if you get the answer, I care that you know how to get to the answer, right? In the last little bit of class, 15, 20 minutes thereabouts, I want to go over some real world examples. And this comes back to that point that I made at the beginning, right? Why are you here? Why do you need this class? Well, biochemistry sounds boring, and it is boring when I just talk at you. But when we try to apply what we learn to real world examples, many of you are going to be doctors, many of you nurses, what have you, right? Why is what we're learning relevant to you, right? There's biochemistry all around us, right? Foods, cosmetics, diseases, medicines, drug discovery, right? There are pandemics, right? There's biochemistry all around us. So what we want to approach this as is really trying to learn biochemistry as a language, right? Do I care that you know every single step in glycolysis? Kind of. What do I care more about is what that means. Where does glycolysis fit in your life, right? Or what you're going to do, right? So today, we're going to take it easy. I'm just going to go over some core concepts. We'll go over some practice problems and call it a day. Chapter 2, again, read the chapter before class. All classes, read the chapter before class. Do the quizzes, right? There is no penalty for getting answers wrong on the quiz. It just keeps asking you questions so that you can reinforce the topics that you learn. The same for the homework. I have no interest in penalizing you for trying different things. There is no penalty for trying the same question over and over until you get it, right? I want you to get it. I don't want to have to, quibble about points. So in theory, there's 25 plus 15, there's 40% of your grade that is free points if you do them. The remaining 10% is split up into 2, 5% class participation, 5% what's this paper, right? We are going to be doing short papers, 1,000-word paper. I don't want to get too bogged down into that right now, right? There's a lot of stuff going on. When the time comes, when you learn some more topics that you can write about, we'll go over what the paper will look like, what I expect from you from the paper. And it shouldn't be onerous. It's 5% of your grade. It should be pretty straightforward. The remaining 5% is that class participation. Not today. I am going to call on people, right? I am going to call on people to come down and do problems or give us some input on a case study, right? You should be in class. You should be ready to at least try to answer a question or to give your perspective on something, okay? All right, so let's get to it. Any questions before we start? Yes, no, good, okay. So, chapter 1, introduction. So what is biochemistry? Biochemistry really can boil down into shapes and energy, right? Two most important topics in, well, really, most sciences are energy, obviously, but specifically for biology, biochemistry is shapes. It's the shape of a molecule that's going to give that shape its function. For example, right? If we look here at these 3 different proteins, these are the same enzyme. It's a tata-binding protein from 3 different organisms. We have one from Arabidopsis, which is a plant, from Homo sapiens, which is human, and we have from an archaea, right? It lives in volcanic areas, right? They all do the same thing. Tata-binding protein is simply a protein that binds DNA at a specific place. What do you notice about these 3 enzymes, about these 3 proteins? Don't get too bogged down about what proteins are made of or anything like that. We're going to cover that on Friday. So what do you notice right away about these 3 different proteins?
They all have the same shape.
They all have the same shape. And they have to have the same shape because they do the same thing. So even though these 3 different enzymes are encoded in the genomes of 3 vastly unrelated organisms, or distantly related organisms. They're likely composed of very different amino acids, like we'll talk about on Friday. Because they do the same thing, they have to have the same shape. It's the shape of a molecule that gives it its function. The same could be true or said for DNA. The shape of DNA, as we'll talk about in just a minute, is uniquely suited to do what DNA is supposed to do. What is DNA supposed to do? What is the job of DNA? Huh? Encode genetic information, and more specifically, how should it do that? It should do that faithfully, robustly, and what's one more important thing that DNA has to be able to do? Repair doesn't have to be repaired. It's good that it can be repaired. But what is one thing that it must do? Be replicated. And it's the structure of DNA that both allows it to encode information, encode it faithfully, encode it robustly, and be replicated. That shape, the structure of DNA. The same is true for RNA and for proteins and so on. All right? Now, that's the structure. Now, thermodynamics, okay, is the other, the energy is the other important part of biochemistry. Really, all thermodynamics and all energy questions boils down to this. Is there enough energy available for this process to take place, for any process to take place? Right? We're just a walking bag of chemical reactions, right? But there's nothing special about us. We are able to move around and think and do all these different things simply because that's the end result of a process at which we are now at the lowest energy, right? If there is not enough energy available to do a process or a reaction or an interaction, okay, that process cannot take place. And this is true of any process, not just chemical reactions, just any process. We'll see that here in just a minute, right? Now, there are many ways to define and analyze both of these, structure and thermodynamics. We're going to go over some of those throughout the semester, right? So thermodynamics specifically gives free energy, okay? There are many ways to define that and quantify that depending on the context, right? Is this a process, is this a chemical reaction, is this an interaction, what have you, right? We're going to go over how to quantify those when we get to those. Now, additionally, biochemistry is defined by interactions between macromolecules and metabolites, right? Macromolecules, like the name implies, are just really big molecules. So these could be DNA, could be RNA, could be proteins, right? Those are three big categories of macromolecules. We also have carbohydrate macromolecules, for example. We'll get to those later, okay? Metabolites are things that are acted on usually by these macromolecules. So these could be our fuel molecules like glucose or sucrose, fructose, fatty acids, okay? Intermediates in a biochemical pathway we'll get to. So these metabolites are acted upon by these macromolecules, and these both exist together in the cell, right? Now, they might be separated by different cellular compartments, but for the most part, they exist together in a cell. And these macromolecules and these metabolites from all different biochemical pathways are acting together. They cross-talk with one another. Metabolites from pathway A might be intermediates in pathway B, or they might regulate pathway B, right? These are all happening at the same time, right? So it's very easy to put everything into a box, right? When we talk about fatty acid oxidation or glycolysis, right? Well, we know glycolysis got 10 steps. We start here and we end here. But we can't forget that this is happening at the same time as glycogenesis or gluconeogenesis or other biochemical pathways. And we don't have to talk to each other. There are some pathways that we only want turned on when other ones are turned off, or vice versa. There are other pathways that we want both to be turned on at the same time. How are these regulated? How do these talk to each other, right? And this gets to the heart of what we are looking at here, types of interactions. And there are four major types of interactions that we're going to be encountering. Covalent interactions, everybody knows those, lovely. Ionic interactions, hydrogen bonds, which are going to be critically important, so I do want to go over those in some detail, and van der Waals interaction. Right, so let's start with DNA as an example. Right, we're going to use DNA as an example to examine these different types of interactions, these different types of bonds. And we're going to go over more detail in DNA later on in the semester. All right, for now, we just want to look at DNA as an example to illustrate these different types of interactions. Okay, so let's look at DNA structure first. Let's just get an idea for how DNA looks. DNA is a macromolecule. Most macromolecules are what we call biopolymers. That's simply just a fancy word for a polymer, right? Made up of biological units. In the case of DNA, those are deoxyribonucleic acids, right? Or DNTPs, the deoxynucleotides. The deoxynucleotide is made up of 3 parts. We have our sugar base, right? Or not base, just our sugar, which in the case of DNA is deoxyribose. In chapter 11, we're going to go over what a sugar actually is and why ribose looks the way that it does. But for now, just know that ribose is this red part here. Right, so here we have our deoxyribose. We call it deoxyribose because it's missing an OH here at this position. Right? Ribose typically has an OH there. In DNA, deoxyribose is missing that hydroxyl. Right, we'll get into why later on in the semester. Right, so here we have our sugar. The second thing that always must be present on a deoxynucleotide is the base. We have 4 possible bases: adenine, thymine, guanine, and cytosine. Okay, these are nitrogenous bases in 2 different families. We have our purines and our pyrimidines. Right? Again, don't get too bogged down in that just yet. But each base is always going to be connected to this carbon on ribose, carbon number 1, or the 1' carbon. It's going to be connected via an N-glycosidic bond. Again, chapter 11 we'll see what a glycosidic bond actually is, but just know that base is always connected to 1' carbon on our deoxyribose. Here's our 1' carbon, 2' carbon, 3', 4', and 5' carbon. Right? So this is how the carbons are labeled on our deoxyribose. The last thing in a deoxynucleotide is the phosphate group, right? So here, for example, we have 1 complete deoxynucleotide with a sugar, a base, and a phosphate. These nucleotides, or deoxynucleotides, are connected together in a very specific way, right? The hydroxyl on the 3' carbon of 1 nucleotide, this one, for example, right? This OH, becomes a nucleophilic attack on an incoming DNTP. It doesn't matter right now. But just know that the 3' carbon of 1 base is bound to a phosphate, and this phosphate is then bound to carbon 5' of the following base. So we have a 3' to 5' linkage. And here we have a phosphoester, and here we have a phosphoester. So we call this a phosphodiester bond. Each nucleotide monomer of this biopolymer is held to the next unit by a phosphodiester bond. And it continues on and on and on. What this means, right, is that a DNA strand is not symmetrical. It has a polarity. On this end, let's say that this is the end of a DNA strand. Well, here we would just have the phosphate connected to carbon 5'. So there would be a 5' end. So we have a 5' phosphate over here. We continue on, all right, let's ignore this phosphate here and say that we end at this side. Well, here we would normally have a hydroxyl. And here we have an exposed 3' carbon, so here we have our 3' hydroxyl, and here's our 3' end. So we have a polarity. We have a 5' end of DNA and a 3' end of DNA, right? We always deal with DNA in the 5' to 3' direction because that's the direction in which DNA is synthesized, but again, we'll get to that later. Additionally, right, so here we have 1 strand of DNA. DNA forms a double helix. And so if we say that this is the red strand here, okay, we have, well, here will be our 5' end, all the way to over here is our 3' end. The complementary strand, right, this complementary strand runs in the opposite direction, right? So instead of 5' to 3' it would be 5' to 3'. So we have one running in one direction one way, and the other running in the other direction the other way. Why is this the case? Well, it has to do with the interaction between the actual bases, right? You might have remembered from Gen Chem or genetics, right? How are these bases between the 2 strands put together? What kind of bond is it? It's a hydrogen bond, right? As we'll see when we talk about hydrogen bonds, we need a hydrogen bond acceptor and we need a hydrogen bond donor, okay? If we were to flip one of the strands so that they're running parallel instead of anti-parallel, well, now the pattern of hydrogen bond donors and acceptors on neighboring bases would no longer match, so they wouldn't hydrogen bond. Additionally, right, remember I told you those nucleotides come in 2 families, pyrimidines and purines. Well, here we can see our 2 purines, adenine and guanine, and our two pyrimidines, cytosine and thymine. We always pair a purine with a pyrimidine, in this case A with T, right, or C with G, right? One purine and one pyrimidine. We never have two purines or two pyrimidines, right? And why is that? Why do you think that might be the case? Right, let's look at, for example, AG. Right, if we look at A and G, right, we'll come back to this later, but I will tell you, right, here we have a hydrogen bond donor, and here we have a hydrogen bond acceptor. Here we have a hydrogen bond donor, here we have a hydrogen bond acceptor, okay? Here we have a hydrogen bond donor and nothing. So, in theory, if we were to take this G, right, turn it around, well now this got hydrogen bond here, and this got hydrogen bond here. So we would get a base pair, but we don't see that in DNA, right? An A never base pairs with G, even though here it seems like it could because the hydrogen bond donors and acceptors would match up. What about these two would stop us from seeing an AG base pair in a double-stranded DNA? Exactly, they're too large, right? Here we have a bicyclic ring with another bicyclic ring. That's going to be wider than the diameter of DNA, which is why we only ever pair a purine with a pyrimidine. So in this case, could we base pair A with C? Well, here we have purine and here we have a pyrimidine, so we say, all right, maybe. But now if we look at the pattern of hydrogen bond donors and acceptors, well, here we have a hydrogen bond donor, here we have a hydrogen bond donor. So that doesn't work. We need a donor and an acceptor. Here we have a hydrogen bond acceptor, here we have nothing. Here we have nothing, and here we have nothing. So we get no hydrogen bonding in between these, so we don't get AC base pairs, and we don't get AG base pairs simply because they're too large. And the same holds true, for example, of CT base pair. In this case, they're too small, right? So they wouldn't meet in the middle, there'd be a gap in between them, so the hydrogen bond couldn't form, right? So we only get base pairing between a purine and pyrimidine, but only if their hydrogen bonds match up, hydrogen bond donors and acceptors match up. So here we see already, right, the structure of the bases are ensuring that we get correct base pairing. And why is that important, right? Well, now we see we can only form AT base pairs, right? Because here we have a purine and pyrimidine, and their donors and acceptors match up. We can never have an AC or an AG base pair, okay? Why does that matter in the scheme of DNA? Try to think about what DNA is supposed to be doing. Now, what is the job of DNA?
It needs to be stable enough so transcription.
Right, it has to be stable. Okay, we'll get to stability in a minute. But it has to be either transcribed or replicated. And now we know, well, if we have an A on one strand, we will always have a T on the other strand. By the same logic, if we have a T, we have an A, if we have a G, we have a C, if we have a C, we have a G. That's always the case. That's enforced, not by some, magical spell somebody put on the DNA. It's enforced by the structure of the components that make up DNA. Additionally, back to your point about stability, we look at covalent interactions, right? Well, covalent interactions is simply the sharing of a pair of electrons between two neighboring atoms, right? It's actually very strong. So carbon-carbon single bond has an energy of about 350 kilojoules per mole, about 12 ATPs worth of energy, right? So they're very strong. However, we can make them even stronger, right? So what is this called here, right? What are we observing here? What is that called? Resonance, right? So which one of these is correct? See, somebody from this side of it. It's been awfully quiet over here. Which one of these is correct? Is it the left one or the right one? Neither, or both.
But it wouldn't be neither. It would be like the resonance structure.
It would be what?
It would be neither.
It would be neither. How could it be neither?
Because it's like a resonance structure.
Huh?
Okay.
Speak up.
It's like there's like a resonance structure where it's in between those two.
Exactly, it's in between, right? What does that mean that it's in between? It means that this one, right, if I were to go ahead and take an adenine, this is what this is, adenine, out of one of your DNA strands, right? I couldn't say if it was definitely here or definitely here. It exists as either one, right? And there we're getting into some quantum shit that I don't even want to think about, right? But either way, right, we get this superposition of both of these equally correct structures, right? The valences of all of the atoms in both of these structures are satisfied. So either one is correct, but neither of them exist. They exist as something in between. What does that mean for the stability of this particular molecule?
It's more stable.
It's more stable. When something exhibits resonance, it is more stable inherently. All right? All of the bases in DNA and in RNA exhibit resonance. Why is this important? Well, again, what is the function of DNA?
Needs to be put together and broken apart, but not broken all the way.
No. Doesn't have to be. It's not nothing to do with being broken. What is the job of DNA? Again, it's to store genetic information, but how? In the bases, yeah, but it has to store it, remember, faithfully and robustly, right? If we had some other base here that didn't exhibit resonance, well, now there's a higher chance that that base could spontaneously either decompose or be acted on, and now we have something that is not supposed to be in DNA. But having a more stable molecule actually hold that information, well, now we are able to more robustly store that information. All right, so if we have, for example, on the left here, right, a mixture of single-stranded DNA molecules, okay, 50% of them have a certain sequence, 50% of them have a different sequence. These are complementary strands, meaning that they should base pair together. What we observe, right, is that they do spontaneously come together. They do spontaneously form a DNA double strand. There is no accessory enzyme or compound or any different temperature that we need to achieve. Well, not temperature, but there is no extra anything that we need in order to achieve these double strands. So this process then we say is spontaneous, right? It will occur. And we'll get to what spontaneity means in just a second, so we go over Gibbs free energy, but this process does occur. Now we went over already the covalent interactions of DNA, right? So it's holding the sugar phosphate backbone together. It's stabilizing the bases. But how are we getting interaction between the two different DNA strands, right? Is it a covalent interaction between the two of those? No, right? There's nothing, the two strands of DNA are not sharing any pair of electrons. So what's holding them together? Well, what's holding them together are these weak interactions. And these weak interactions are of critical importance in any kind of interaction that we talk about in biochemistry. Here, in the case of DNA, but the same is true when two proteins interact, or when a protein interacts with a substrate, or when two cells interact, right? These are almost always mediated by weak forces, right? So let's go through the weak forces that we're going to encounter this semester and see how we can quantify them and what they look like. First of which, the strongest non-covalent interaction being the ionic interaction. Right? So this is very easy to understand, very easy to conceptualize, and it's also the strongest weak interaction. Here, simply, when we have two opposite charges near each other, they attract. When we have two of the same charge near each other, they repel. Right? So just like a magnet. We have north and south, they attract. When we have two north or two souths, they repel. Right? So it's exactly the same thing going on here, but we can quantify the magnitude of that interaction. Right, using this equation here, E equals K Q1 Q2 over dr. So E is simply going to be the energy. It's going to be the energy of that interaction. Q is the electronic charge on each of the atoms or molecules in question in units of electronic charge. So we don't have to futz around with converting them to coulombs or anything. For example, if we have a sodium and a chlorine ion, well, the Q on the sodium is +1, and the Q on the chlorine is -1. Easy peasy. That's what this K allows us to do. This K is the proportionality constant. This is just a constant of the universe, and it's been devised such that we can use the electronic charge, and here at distance intention, we'll get to that in a minute. And for our case, it's 1389, right? 1,389 kilojoules per mole, or kilojoule angstrom per mole, right? And this is just gonna allow all the units to cancel out and get us an energy that actually makes sense, right? D is the dielectric constant, right? So in a vacuum in space, the interaction between two charged particles is just what it is based solely on the distance in between them because nothing's getting in the way. But all of our reactions aren't happening in a vacuum. Where is most of our reactions going to occur? Let's see, somebody else. Someone hasn't spoken today. Where are all of the reactions we're going to encounter? Where are they going to take place?
In air or in a solution.
In what?
In air or in a solution.
Say louder.
In the air or in a solution.
Well, not in the air, but yes, in solution, right? And what kind of solution? Huh? The cytoplasm, all right, what's it mostly made out of? Water, exactly. It's going to be taking place in functionally, it's just the same as water. So now we have all this water in between our two particles, so the interaction is going to be attenuated. That's what the dielectric constant tells us. In water, the dielectric constant is 80, meaning that the interaction between two charged particles is going to be 80 times weaker in water than it would in a vacuum. That makes sense, that's fine. And then lastly, R is just the radius, or the distance between our two charged particles. The important thing here is that the distance must be in angstroms, right? Not nanometers, not micrometers. It must be in angstroms, otherwise this proportionality constant doesn't work, right? So just make sure that your distance is in angstroms, your charge is in units of the electronic charge, and this tells us now that, or what the magnitude of the interaction is between them, right? So, if we have a positive and a negative charge particle close to each other, what is the sign going to be of the energy? Negative, right? So if the energy between two particles is less than 0, then we have an attractive force. Now what do we have? Two negative charges next to each other. Negative times a negative is positive, so E will be positive, will be greater than 0. If the energy between two charged particles is greater than 0, that's a repulsive force, right? That's a definition. What does it mean if the energy is 0? If there's no charge, there's no energy, which means that there must be at least one of these charges 0, right? If we have a -1 and a 0, well, this is on top, so all that cancels out to 0. So we can only have, of course, ionic interactions with an attractive repulsor when both of the interacting partners have some kind of charge. Right? Now, how does this matter in DNA, and how does DNA illustrate this? Well, remember the structure of DNA. We have our sugar phosphate backbone and the bases. So let's ignore the bases because they don't have a charge. Okay, so they're not going to have any ionic interaction at all because the charge is 0. The sugars also we can ignore because they have a charge of 0 as well. But what doesn't have a charge of 0 are the phosphates in between them, right? The sugar phosphate backbone. So if we look here, right, here we have one strand, here we have a second strand, right? What does this tell us? Well, here, these purples, these are the phosphates. Let's look at this phosphate. Well, this negative phosphate is now interacting with the phosphates on the corresponding or complementary strand. And because they're both negative, what's the sign of the energy going to be? Positive. So it's going to be an attractive force or a repulsive force? Repulsive force. Do we want that? What's going to happen? The two strands are going to not want to be together, which is no bueno. So instead, what do we have to do? Well, ignore what's the correct answer, but imagine that you were omnipotent and you can do whatever you want to any chemical structure. How do you get rid of this repulsive force between the phosphates? Let's say you can change the laws of physics. You can do whatever you want to do. How do you get rid of this repulsive force? Make the phosphates less negative, or make it positive, or just get rid of the charge altogether. Now we can't do this obviously, so instead what does this cell do? Well, the cell uses these cations, like sodium, magnesium, potassium, these positively charged ions. What's going to happen? Let's say that we have magnesium chloride in solution with our DNA, right? Well, what's going to happen now? Well, that magnesium is going to dissociate into Mg2+, and we'll get 2 chlorines as well, right? So what's going to happen now? Well, now the chlorines, which are negatively charged, what's going to happen if they get close to the DNA? What's going to happen if they get close to the DNA? The Cl minuses.
Would they break the repulsion between the phosphates and the positive ions?
Well, how can they get there, right? If the phosphates are negatively charged, the chlorines are negatively charged. So what's going to happen with those chlorines? They're just going to be pushed away. They're not even going to see the DNA. However, the magnesium or the sodium or the potassium, whatever, those are positively charged, and they will have an attractive force with negatively charged phosphates. So they're effectively going to coat the outside of the DNA. And what's going to happen now? Well, now if we examine not this phosphate rather, or this sodium, but instead we examine this region of space, well, now the charge in that region of space is effectively 0, or close to it, which means that the Q changes, the E goes down closer to 0, or up, down closer to 0, and gets rid of these interactions. We have to remember this is happening here with all of these phosphates. This phosphate is interacting with these, this phosphate is interacting with these ones are interacting with all of these. So this builds up, right? So we must have a way to ameliorate the repulsion between those phosphates on the backbone. In fact, that's one of the reasons that salt is so important. Without sufficient amount of salt, your DNA duplexes will slightly get destabilized. This is more apparent in the lab, right? If you have a solution of DNA devoid of salt, it's much easier to pull those 2 strands apart. Instead, when you add salt, those 2 strands anneal or are bound together more strongly. Now, hydrogen bonds don't necessarily hold the 2 strands together, right? Instead, what the hydrogen bond between the bases do is it ensures specificity. It ensures that when we have an A, we can only have a T, and when we have a G, we can only have a C. So let's examine hydrogen bonds in a little bit more detail first, right? So a hydrogen bond is always made up of 1 hydrogen bond donor and 1 hydrogen bond acceptor. The hydrogen bond donor is simply where we find the hydrogen atom. In the donor, where we have the hydrogen atom, that hydrogen has to be covalently linked to an electronegative atom. Now, this class is very simple. That electronegative atom is most always going to be either nitrogen or oxygen, right? So here we have an NH bond, and here we have an OH bond, right? Either one of these are a hydrogen bond donor. Why is that? Well, if we look at this NH bond, well, N is significantly more electronegative than hydrogen. So what does it do? It pulls electron density away from the hydrogen, giving the nitrogen a partial negative charge and the hydrogen a partial positive charge. Now we have the makings of an electron dipole, effectively, right? If we put that near to another electronegative atom, well, now our acceptor, because it is electronegative by its nature, now we have a partial negative on that as well. So we have a partial negative in the acceptor next to a partial positive in the donor, and now we get attraction between them. It's a special type of ionic interaction, positive next to a negative, so the energy will be less than 0. Okay, it's just a special type of ionic interaction, really, you could say. Now, the important thing about hydrogen bond is that the bond angle is actually critical to how strong that hydrogen bond is. The strongest hydrogen bond has a bond angle of 180 degrees, meaning all 3 atoms, the 2 atoms in the donor and the 1 atom in the acceptor, must all be in more or less a straight line. If we deviate from that more than, say, about 5 degrees, the energy of this hydrogen bond drops off quite quickly. In fact, if we look here at the GC base pair, well, again, now we can see, here's our hydrogen bond. What? Is this our donor or acceptor? Shout it out. Donor or acceptor? Here. Donor, exactly. Here we have acceptor. Here we have donor, acceptor. Very good. And we have 3 hydrogen bonds in a GC base pair. And notice we have a straight line between NH and O, a straight line between NH and N, a straight line between OH and N, right? Straight lines all the way across. The same is true for an AT base pair, which which notably only has 2 hydrogen bonds, making GC base pairs 10 more strong. Either way, right? Notice that we have our structure here. So again, the structure of the bases, A, enforces the correct base pairing. They are stabilized by resonance. The structure allows the hydrogen bonds to form so that we have 180 degree or thereabouts bond angles, right? So we see over there, the structure of what's making up DNA is critical for what DNA is supposed to be doing. So here we see Watson-Crick hydrogen bonds, right? So why do we call them Watson-Crick hydrogen bonds? Because those two are the ones that got the name of the Nobel Prize. There's also Rosalind Franklin who did most of the work. That's a story for another day. Either way, that's the name that stuck, the Watson-Crick base pairs. So what we see here are Watson-Crick base pairs. There are only 2, A and T, or G and C. They always look exactly the same, right? Now, because there's no net forming of bonds, there's no energy released, right, when a base pair forms. And let's see what we mean by that. Let's see what we mean by that. So here we have a base on strand 2, and here we have a base on strand 1. We're only looking at the small part of the base, right? So we don't even know what these bases are, but what we do know is that on this base on strand 1, we have a hydrogen bond acceptor here, and over here we have a hydrogen bond donor on strand 2, and these two are supposed to base pair. And, you know, going back to high school biology, right? Whoa, yeah, you know, the hydrogen bonds between the base pairs, they hold the DNA together. This is lies, right? The hydrogen bonds between the base pairs do not hold the 2 strands together. Instead, they enforce the specificity that I told you about before, A with T and G with C. And why don't they do anything to hold the 2 strands together? Well, because when we form a base pair, no bonds are made and no bonds are broken. Remember back to everyone's favorite class, Gen Chem. When bonds are made, what does that do in terms of energy? Does it require energy or does it release energy? It releases energy. When bonds are broken, then the opposite is true. It requires energy. So if bonds broken at the beginning equals bonds formed at the end, well, then it's all a wash, right? We haven't done anything to the energy in the system. It's no more stable than it was at the beginning. So we see here, this hydrogen bond acceptor on strand number 1 in aqueous solution is not all by itself, right? If this was in a vacuum, then absolutely, that would be the case. We'd be forming a hydrogen bond that would release energy. But in reality, the hydrogen bond acceptor is already in a hydrogen bond with the water in the environment, right? And remember, water has 2 hydrogen bond donors here, one here and one here. So here we have a hydrogen bond, and here we have a hydrogen bond with the water on strand number 2. So now, what do we have to do in order to get this guy to interact with this donor here? What do we have to do? We have to break the hydrogen bonds, right? That's going to require energy. That's okay. All right, so here, now we form this hydrogen bond here. So we've broken 2, but we've only formed 1, right? So will this process continue? Will this be spontaneous? Will this happen on its own? We're breaking 2 hydrogen bonds, we're only forming 1 hydrogen bond. No, right? We're breaking more bonds than we're making. But this does happen spontaneously, like we observed back here, right? This does happen spontaneously. So where, you know, what's going on here? Right? We're breaking 2, we're forming 1. But that can't be the case. What has to happen? Well, we could form 1 more hydrogen bond, and then it would be a wash, right? But then at least it would happen. What other hydrogen bond can we form? Exactly, between the waters. So here, yes, of course, we're going to get a hydrogen bond, Watson-Crick hydrogen bond, but the 2 waters that are released, here we have a hydrogen bond acceptor and 2 donors. Here we have an acceptor and 2 donors as well. So this donor can interact with this acceptor, and there we go. We formed 1 more hydrogen bond. So we've broken 2 at the beginning and formed 2 at the end. So this process has a delta G of approximately 0. So what this does, so it's not going to hold anything together because this is not any more stable than this was. This is not any lower in overall energy than we were over here. There's So instead, what the hydrogen bonds do are enforce the specificity, right? Okay, so moving on now to van der Waals, which is always the hardest for people to get their minds around. So van der Waals interaction is the weakest of all of the non-covalent interactions. In fact, if we have any of the other interactions, ionic or hydrogen bonds, they're orders of magnitude stronger than van der Waals interactions, so they're not even mentioned, right? Outside of very special circumstances. So what is a van der Waals interaction? Well, let's consider at time 1, okay, we have two nonpolar molecules next to each other. Uncharged nonpolar molecules, right? So they shouldn't have any ionic interaction, and they don't have any hydrogen bond donors or acceptors or anything like that. So there should be no interaction between them. But what we find is if left alone for long enough, they will start to come together, right? Up to a point. And that point is the van der Waals contact distance. So what is the force acting on them if there is no ionic or hydrogen bond force going on? Well, that's van der Waals. Because though we'd like to think of living in a perfect world where the charge distribution around both of these nonpolar, uncharged atoms, or whatever, are just perfectly symmetrical and spherical all the way around, right? Like we see in T1. So there's zero net charge, and that no net charge is evenly distributed, that electron density is evenly distributed around the entire molecule or atom. But what actually occurs is that every so often there will be a slight asymmetry that arises, right? So at time 2, we see that slight asymmetry. Parts of that molecule or atom will become slightly, ever so slightly negatively charged, but because the overall charge isn't changing, there must be a corresponding change in the charge of the opposite side on the other side, or somewhere else. So we can have a little bit, ever so slightly more electron density over here, which means we'll have less over here, giving this side a partial negative and this side a partial positive. When this happens nearby another, say, atom, this partial positive charge, what's it going to do to the electron density over here? Here we have a partial positive. So what is it going to do to the electron density of the blue object? Yeah.
The negatives from the other side.
Yeah, it's going to attract some negative charge to over here, right? So that's what we see here. It attracts some negative charge, but again, because the overall charge isn't changing, that means that there must be a corresponding change in charge of the opposite sign somewhere else, right? So the overall charge is still 0. But now we have a very slightly partial positive next to a very slightly partial negative, and we get an attractive force, right? And this force is greatest, or has the lowest energy, at the van der Waals contact distance, right? So you might have seen, for example, here, right? Here we see a space-filling model of a cross-section of DNA, right? So notice each of these spheres is an atom. Here we have the phosphates on the backbone. Here we have phosphates on the backbone. Notice that they have different sizes, right? Here we have hydrogens. Here we have the phosphate, right? We have some oxygens over there. Okay, they have different sizes. The sizes of the atoms in this space-filling view is the van der Waals contact radii, or the van der Waals radii of those atoms. Why is that? Well, because if we imagine, well, it gives us an idea, it gives us a rough estimation or approximation of how quote-unquote big an atom is, right? An atom isn't necessarily that big, but it gives us an idea. It makes it easier for our monkey brains to understand, right? So if we try to push two atoms together closer than their van der Waals contact radii, well, now the amount of energy required to do so increases so much that they start to repel each other, right? So we can think of that van der Waals contact radii as kind of the size of that atom. Conversely, because here we have the lowest energy, right, to pull them apart, well, now that energy increases, so we're going to have to put in that energy. If I'm just pulling them apart, that energy is coming from my muscles and from the fuels that I initially ingested, right? But that means that the lowest energy is when they're touching each other, if they're nearby. And why does this matter for DNA? Well, this van der Waals is responsible for what we call base stacking. If we look at the base pairs on subsequent rungs of this DNA ladder, they're more or less parallel, right? What does that mean? Or how are they parallel? Well, they're parallel because of van der Waals interactions, right? say here I have, you know, a base pair, right? So here I have one base, here another base, here another base, and here a fourth base, right? If they're stacked on top like this, parallel, well, now what happens if this one tries to tilt up? Well, these are now too close together, right? And these ones are now farther apart, right? The ones on my fingertips. So that energy has to come from somewhere, and the energy has to come from somewhere to push these two back together. This is a less stable configuration than just having them be parallel. So these van der Waals interactions are important for, in this case, base stacking, but they're also important for other types of interactions like we'll see, specifically protein-to-protein interactions sometimes, right? And then lastly, we have the hydrophobic effect. And I didn't